Hello, everyone. Before I begin my talk, I would like to congratulate the organizers of 6th Digital Congress on Nanobiotechnology and Bioengineering. Looking to the today's world scenario, we find that we have three important problems: population explosion, climate change, and the food security, which is the basic need for the human being. In this context, I have selected bio-based green nanomaterials for plant hair and opportunities and challenges. Well, my name is Mahendra Rai, and during the period of 30 minutes, I am going to cover the population explosion and how we can secure the food for this big population. What are the innovative solutions for growing crisis and how nanobiotechnology can be a game changer in agriculture. Eco-friendly nanobiotechnology by using green bionanomaterials, green synthesis by plants, bacteria, algae, and fungi. And the role of smart nanotechnology for early detection of the disease, delivery of chemicals, and for protection by delivering chemicals. In this talk, I am going to cover a preliminary study on soft rot of ginger, which I did during COVID period. Most importantly, toxicity issues would be discussed and what is the role of the consumers, how they perceive this technology, and what are the safety rules. If you take the stock of the population of the world globally, it is increasing with a very fast pace. In 2011, it was only 7 billion. It increased to 8 billion in 2024, and by 2050, it will be 9.7 billion. So, how to feed such a big population is a question uh, before all of us. This is one side, and on the other side, if you think that the chemicals are increasing with a very fast rate, also, people are using so much chemicals. In 2018, it was 223.7 US billion dollars. And by reaching 2025, it is estimated to be $308.4 billion of purchase of the chemicals. This suggests that there is an increase of $85 billion, such a huge increase of the chemicals, which are really responsible for creating pollution and generating climate change. One would be astonished to know that 1.5% of the greenhouse gases are increased by using chemical fertilizers. So is the case with fungicides and bactericides, which increase from 393 to 530 million kilograms from 1990 to 2018. Such a huge figure. The European Commission has taken a step under the Farm and Food program to reduce the pesticides, nutrient losses, and the use of antimicrobials, for example, bactericides, fungicides, and pesticides, by 50%. It's a good decision. By 2030, there will be the reduction of 50% of chemicals. On the other hand, the organic farming would be promoted and it would be 25%. So this is a novel idea for sustainable agriculture production. In the current state of control strategies or conventional control strategies, there are three strategies: chemical control, cultural control, or crop rotation, or biological control. Using chemical control, we are developing the resistance among the pests, insect pests, and even pathogens, because we use a huge amount of fungicide and pesticides. Even the cultural control is not feasible because it has been observed in pea diseases, pea wilt diseases, the Pisum sativum diseases, that even after 10 years, the mycelium remains in the soil. So, what you can do? Considering these facts, people started considering the biological control methods in which we use certain microorganisms to control the disease, or we use plant metabolites for the control of the diseases. As far as the new approaches are concerned, there are three new approaches. One is gene editing. In gene editing, we use CRISPR-Cas9 technology. By this technology, we can increase the resistance of the host plants against the pathogens. Second is nanobiotechnology or nanotechnology. In this technology, we can use nanoparticles as nanocarriers for the delivery of chemical pesticides or fungicides. By machine learning, we can predict the plant pathogen infection or early detection of the disease, and detecting the plant pathogen would be very useful for the treatment of the diseases. If we go back to 1960, we find that Norman Borlaug gave the idea of green revolution. And uh, in 90s, there was gene revolution after the discovery of PCR. And at the turn of 21st century, that is the nano revolution has been very important factor. And now we can say that 21st century would be guided by nanotechnological principles. Now, if we try to understand what nanomaterial is, the creation of functional materials, devices, and systems at 1 to 100 nanometer can be done. And this will be responsible for different functions. And that is why it is called as enabling technology. If you change the shape and size of the nanoparticles, the activity changes. The smaller particles have a larger surface area. For example, if you take 1 centimeter into 1 centimeter, the surface area is only 6 centimeters. Whereas if we take 1 millimeter into 1 millimeter, it becomes 60 centimeters square. In case of nanometer, if we consider 1 nanometer, this will form the area of 60 million nanometers, such a huge area. And this area is responsible for a broad activity. And that is why I say that nanoparticles are very, very active. They are bioactive materials. And nanomaterials are of three kinds, which we can use in agriculture: organic, inorganic, and carbon-based. The organic nanomaterials can be micelles, liposomes, dendrimers, cyclodextrins, whereas inorganic nanomaterials are silver, gold, aluminum, cadmium, copper, zinc oxide, and copper oxide nanoparticles. The carbon nanoparticles or carbon nanomaterials include the graphene, fullerene, carbon dots or carbon nanotubes. The carbon nanotubes can be of multi-wall or single wall. The principle of green synthesis was first given by Paul Anastas and John Werner in 1991. And according to this principle, the method of synthesis should not involve the harmful chemicals, and it should reduce or eliminate the use of generation of hazardous substances. And Paul Anastas and John Werner wrote a very famous book, Green Chemistry, in 1998. It was published by Oxford University. This book is based on 12 principles of green chemistry. So, taking into consideration the green principle of synthesis, the nanoparticles can be synthesized by green methods or biological methods, which involve fungi, bacteria, actinomyces, algae, and plants. The green materials are very important because chemical synthesis or physical synthesis involve the use of chemicals and which may be harmful, sometimes may be toxic. By physical synthesis methods, sometimes we use very high temperature, say for example 1200 degrees Celsius, and very high pressure. So green synthesis is an important synthesis. In an experiment, we analyze the mycosynthesis, photosynthesis, and chemosynthesized nanoparticles. And we evaluated against E. coli and Staphylococcus aureus. It was found that the mycosynthesized nanoparticles by Fusarium orbiosporum demonstrated the minimum nanoparticles, that is minimum inhibition concentration compared to synthesis by plants, that is as a direct indica or chemically synthesized nanoparticles. The chemically synthesized nanoparticles demonstrated maximum minimum inhibitory concentration. That is to say, maximum MIC for checking the growth of E. coli and S. aureus. The green nanomaterials are eco-friendly, economically viable, biocompatible, and very, very active against the pathogens or different microorganisms. They can be synthesized by algae, fungi, bacteria, actinomyces, and plants. Fungal system is very important, very useful because fungi have high tolerance towards the metals and high wall binding capacity. These organisms are easy to culture on large scale, can be synthesized by extracellular methods. Their downstream processing is also very easy. So fungi plays a major role in synthesis of nanoparticles. Based on these facts, in 2019, we gave a new concept of micro nanotechnology, and it was coined by our group. We proposed this idea with Paul Bridge of Cabby, Commonwealth Agriculture Bureaus of England, and it was published in Applied Mycology. Micro nanotechnology is nothing but use of the fungi for synthesis of nanoparticles and their applications in agriculture, medicine, or uh, as a catalyst. We screened about 15 Fusaria species under the Indo-Brazil program. We had a project and we evaluated 18 Formica species for synthesis of silver nanoparticles. Besides this, Alternaria species, Aspergillus species, mushrooms, and Candida species were also evaluated for the synthesis of nanoparticles. And finally, we came to the conclusion that fungi plays a crucial role in nanoparticle synthesis and producing the green nanomaterials. Here is an example which I would like to demonstrate here. Fusarium culture, Fusarium of acuminatum and Fusarium solani were isolated from ginger and onion, respectively. And we synthesized the silver nanoparticles of 5 to 40 nanometers. We prepared the extract of these fungi and treated with 1 millimolar silver nitrate. And the nanoparticles are very, very bioactive against different pathogenic fungi. Among the fungi, Fusarium we have evaluated, we found several nanoparticles of silver. And here you can see some of the fusarium species: Fusarium graminearum, Fusarium solani, Fusarium oxysporum, Fusarium calmorum, Fusarium skirpi, Ricinctum. Among these species, Fusarium oxysporum was found to be one of the best species because of the synthesis of smaller nanoparticles and more bioactive ones. The hypothetical mechanism of the synthesis of silver nanoparticle was for the first time proposed by Nelson Duran of Institute of Campinas. He proposed that NADPH and nitrate reductase and anthraquinones are responsible for synthesis of nanoparticles, for reduction of silver to silver nanoparticles, and the electrons are taken from NADPH, nitrate reductase, and anthraquinone. We also tested nitrate reductase enzyme. We treated the extract of the fungus with the disc of nitrate reductase, and we found that silver nanoparticles are formed. Whereas in case of only disc and distilled water, there was no silver nanoparticle, or silver nanoparticles were not produced. When the proteins on the caps of nanoparticles was analyzed, it was found that the capping is due to acid phosphatase enzymes, glycosidase, 1,3-beta glucurosinyl transferase, and serine carboxypeptidase. These are the enzymes, proteins responsible for capping of silver nanoparticles. And these cappings are attached to the silver nanoparticle with the sulfidal groups. All these proteins are involved in metabolic pathways of fungi and belong to hydrolytes. In agriculture, we can use the nanoparticles for plant protection as well as for plant growth promotion. For plant protection, we can use nanoparticles for early detection of diseases by forming nanosensors, or we can directly treat the pathogens by nanoparticles. On the other hand, we can use the NPK in nanoform or other fertilizers which can be used as nanofertilizers for growth promotion. There are several abiotic and biotic factors responsible for uh, the crops. Sometimes abiotic factors are deteriorating. On the other time, biotic factors or together also they can affect the life of the plants. The abiotic factors like heat, cold draw, flood, and salinity, they can destroy sometimes the plants and the crops. Among the pathogenic microbes and pests, fungi, bacteria, viruses, phytoplasma, insects, aphids, and so on are responsible for deterioration of the crops. The early detection of the plant disease can be done by nanobiosensors, and nanobiosensors can be developed by using the protein capped with nanoparticles or nanoparticles uh, which are capped with the proteins, and they can recognize several analytes like nucleic acid, antibody, and proteins, transfer this signal to optical, electrochemical, mechanical, and magnetic transducers, and from transducers, they can be transferred in the form of signals, and we can measure these signals and detect the disease. If there is early detection, we can start the treatment of the pathogens as early as possible. The nanoparticles against plant pathogens can be used for killing the pathogens or as a nanocarrier for applying on the pathogenic microbes. It can be said that there is a direct nanoaction, or they can be killed by antimicrobials, and the nanoparticles can be used as nanocarriers. These nanoparticles can be applied on leaves, that is foliar application, or into the roots so that they can be transferred to other parts of the plants. These nanoparticles include silver, gold, copper, titanium oxides, silicon oxide, iron oxides, nickel oxide, cesium, and carbon nanotubes of multicellular, multi-wall, and single-wall carbon nanotubes, magnesium hydroxide nanoparticles, or fullerene, graphene, or carbon dots. So all the nanoparticles, they are really very, very effective against treating the plant diseases. I want to give you the example of ginger here. And this study was carried out during the COVID period. I was in home quarantine and I thought of what to do now. And then I conducted one experiment. As you know, India is the largest producer of ginger. China and India contribute 50% of the ginger production in the world. From 2015 to 2016, according to a data, 800,000 tons of ginger is produced by India. In 2023, it was 2.4 million tons. And ginger is being used in India and other Asian countries because of their medicinal properties. They are antimicrobial, anti-inflammatory, antipyretic, antioxidative. They can be used against diabetes, for protection of livers, diuretic, and of course, highly anti-cancer. So 50 to 60% of field loss is there. So how to combat on these kind of pathogens which are responsible for half of the crop production? We isolated two fungi from the ginger deteriorated plants and the soil. These are Fusarium and Pythium species. These species are responsible for soft rot of disease. Sulfur nanoparticles and copper nanoparticles were synthesized, and we applied these on ginger rhizome, and we found that sulfur nanoparticles demonstrated after 90 days the maximum growth of the plants of ginger, and copper is lesser than the sulfur nanoparticles. And the positive control of redomyl gold also demonstrated protection of the plant from soft rot disease. Whereas in control, it is observed that plant pathogens destroyed these plants. We evaluated chlorophyll A fluorescence by using NDP, and this equipment is very important in demonstration of chlorophyll A fluorescence. It was found that plants which was not treated had shown no fluorescence, almost no fluorescence. It can be observed, and here uh, in the red dots, whereas the blue one, it is copper nanoparticle treated plant. It was found that it showed very high fluorescence, followed by redomyl gold and sulfur nanoparticles. As a matter of fact, sulfur nanoparticle demonstrated the maximum growth of the ginger plant, which suggests that sulfur nanoparticles are very effective. We also analyzed the effect of sulfur and copper nanoparticles by radar plot, and we found that the performance index, which we analyzed, was maximum in case of sulfur nanoparticle. Here it has been shown by orange lines, followed by copper nanoparticles and redomyl gold. Whereas in case of control, it has been designated by red here. Uh, There was no fluorescence. The performance index was zero. On the other hand, the electron flow was also very high in case of sulfur-treated nanoparticles, followed by copper-treated nanoparticles, whereas in case of untreated nanoparticles, that is designated here with red line, there was no performance index. The effect of sulfur nanoparticles is more clear by membrane and leaf models. These are called as pipeline models. Here, the performance index is only 2.51, whereas in case of control positive, that is to say, when the plants were treated with readable gold, it was 10.107. In copper treated nanoparticles, it was almost 9.77, or you can say 10, whereas in case of sulfur nanoparticles, it was maximum 13.42. So it is clear. Here also, if you observe the reaction center, all the reaction centers are closed in case of untreated plants, whereas in case of redomal gold treated plants or fungicide treated plants, there are only some closed centers, whereas other centers are open. This suggests that copper nanoparticles and readable gold are similar in effect. In case of sulfur nanoparticles, there is not even a single reaction center which is closed. All the reaction centers are open. And that is why the performance index is higher. Now, coming to the nanofertilizer, if we compare the conventional nanofertilizer with the nanofertilizer, conventional fertilizer and nanofertilizer are entirely different. In case of nanofertilizer, they have high solubility, bioavailability, they are controlled release, and very effective duration. Just opposite to nanofertilization, in conventional fertilization, you can see they are lesser soluble, lesser bioavailable, there is no control release, and so on. We can use very less input of nanoparticles as fertilizer, whereas in case of conventional fertilization, 100 to 782 kilogram per hectare is used. And unfortunately, 50 to 90%, 80 to 90% is lost due to leaching. And that goes to the soil and sometimes responsible for eutrophication of the lakes. Nanofertilizers can be of three types. Nano-range fertilizer, that is nanoparticle as nutrients, or nano-range additives, which can be used in traditional fertilizers, along with the traditional fertilizers, and nano-range coating. The traditional fertilizer can be coated with the nanoparticles. So these all are very effective fertilizers. They can be applied uh, by in vitro and in vivo methods. In vitro, they can be applied by aeroponics or hydroponics. In foliar application or soil application, in case of vivo, mostly foliar application is much more effective. A simple experiment was performed in my laboratory, and we used maize seeds and treated with magnesium hydroxide nanoparticles. We selected magnesium because magnesium forms the phytol ring. It is one of the important element of phytol ring, which is responsible for chlorophyll. So different concentrations were applied, and it was found that 500 ppm is most effective on germination of maize seeds as well as their growth. These nanoparticles were synthesized by Aspergillus niger. And then the nanoparticles were mixed in the miracis and spooks medium, and then maize seeds were grown. Miracis and Scoop's medium is a medium which is used in plant tissue culture for growing the seeds or plant organs and tissues. And in other experiments, we treated the seeds first and then inoculated into the medium. In both experiments, we found that 500 ppm of nanoparticles is much more effective. We also experimented in pea seedlings. These are pigeon peas, which are common in India and used in vegetables and food. These pea seedlings were the seeds were treated with 20 ppm nanoparticles of 20 to 30 nanometers. It was found that the treated uh, plants with copper nanoparticles were taller than the untreated plants. So if we consider the studies, different studies made as fertilizers on different plants, we find that so far titanium oxide, cerium oxide, silver, copper, iron, cesium, carbon nanotubes, carbon dots even, zinc, and silicon oxides, magnesium, manganese, NPKs, and magnesium nanoparticles have been used against different crops like soybean, chickpea, maize, wheat, tomato, bean, rice, lettuce, barley, watermelon, and so on. So there are a number of crops which have been treated by metal and non-metal biodegradable nanoparticles. Now, toxicity is a very important issue, and everybody is concerned about the toxicity to the agriculture, environment, and to the human being. It is a very interesting study. Uh, in this study, the fertilization by conventional nutrients and nanonutrients is compared. The conventional nutrients show more ammonia emissions as compared to nanonutrients. Here, as far as the nitrous dioxide is concerned, it is much more in case of conventional nutrients, whereas it is less in quantity when nanonutrients were used. And finally, sometimes these are mixed into the lakes and they are responsible for eutrophication of lakes, where phosphorus and nitrogen is increased, and they give rise to a different kind of vegetation, and the lake becomes shallow. So, how to avoid these? Another experiment we did with Ann Anderson of University of Utah of United States. We selected Pseudomonas putida, very good bacterium. It is a rhizosporium bacterium. And we developed the biosensors with the help of Lux AB gene. And when these bacteria were treated with 5 to 12 nanometer size silver nanoparticles at concentration 0.4 microgram per ml, they were died. And we tested it in biosensor. These nanoparticles also penetrate into the membrane. And here you can see all the nanoparticles, they go inside. Of course, it depends upon shape and size of the nanoparticles because the small size nanoparticles have got more entry compared to the bigger size nanoparticles. And they destroy, when they go inside, they destroy mitochondria and disrupt the respiratory chain, and respiration is totally deteriorated. On the other hand, they are responsible for checking the multiplication of DNA. And there is no multiplication of DNA when it comes in combination with the nanoparticles, particularly silver nanoparticles. So finally, I would like to say that toxicity is a very important issue, but it depends on shape, size, concentration, and surface coating of the nanoparticles, and sometimes also aggregation, because in case of aggregation, the bioactivity decreases. And if bioactivity decreases, the pathogen is not killed. There are various challenges before us, and therefore more studies concerning toxicity and safety is required. More safety uh, rules should be formed. There are unforeseen risks because we have not studied. A thorough studies is required to fight with the unforeseen risks. There are various ethical issues, and most importantly, public acceptance of the technology is needed. The consumer's role here is very important. For this, we need to organize the meetings to develop awareness, not only awareness, but attitude and skills among the common people. And finally, the effective regulatory mechanism is important. One can form the rules, but sometimes rules are not implemented. Most important is to implement these rules, and then only we can combat the toxicity issues. There are various concerns like metal nanoparticles are not biodegradable. Even the synthetic polymers are not readily biodegradable. And the toxicity of nanoparticles, especially the smaller nanoparticles, are very toxic. And therefore, if there are problems, there are solutions as well. We should go for biodegradable nanoparticles and comparatively bigger nanoparticles because they are safer as compared to the nanoparticles, which are very small. Here are some of my recent publication of the books: Nanotechnology in Plant Health, Nanophytopathology, and Nanotechnology for Environmental Management, for Nanotoxicity in Agriculture and Environment. So I hope these books may be important for you. I would like to acknowledge different funding agencies. Foremost is Federal University of Piaui, UFPI in Teresina, and my own university, Sant Gadge Baba Amravati University in India. I have police collaboration as well. I'd like to thank NAVA, CNPK, University Grant Commission, New Delhi, and Department of Science and Technology, Government of India. So, muito obrigado. Thank you for patient hearing. Muchas gracias. Cinque. Dhanyavad.